Chad is a fourth-year medical student from Northwestern School of Medicine. Chad will be discussing his work with Dr. Jayabalan, who's also here today, when I heard, on metabolomics. So they're looking in the investigation of difference in metabolic markers in walking adolescents and young adults with cerebral palsy compared to those without. So their team's looking at using biological markers to develop novel, individualized rehabilitation regimes and exercise strategies for individuals. So thank you, Chad and Dr. Jayabalan for coming today.
Hello, good morning. Good morning, everyone. Thanks so much. It's a true honor to be here to share our work. And on behalf of Dr. Jayabalan, Dr. Dayanidhi, and the rest of our team, we're really excited to share the work that we've been doing on sort of a bigger project looking at adolescents and young adults with cerebral palsy, and specifically looking at biomarkers. But today we're going to be really focusing on altered systemic metabolomics in ambulatory young adults with cerebral palsy. I have nothing to disclose. Sorry, there's... The animation didn't go through, but if you can see through this here, I'm going to kind of explain the text that you should be able to read behind this banner here. But essentially, so cerebral palsy manifests across a wide spectrum of functional capacities as characterized via the Gross Motor Function Classification Scale, or GMFCS, ranging from 1 to 5, from least to most functionally impaired. And even in those who are able to ambulate or walk independently, it has been shown that individuals with cerebral palsy spend more time in sedentary activity or are less active than the general population. And it is also associated with higher rates of numerous comorbidities or concurrent medical conditions, such as things like diabetes, high blood pressure, asthma, osteoarthritis, osteoporosis, and a number of physiologic abnormalities or alterations in the way that the body at the cell level operates to carry out function. And some of these things include altered cardiometabolic health, so like I mentioned, higher blood pressure, inadequate nutrition intake, as well as higher energy costs with activity, so requiring greater energy to perform certain functions, as well as reduced muscle mitochondrial content and activity with disuse. And so if you remember back to grade school science, the mitochondria is the organelle within the cell that helps to produce energy. And so in cerebral palsy, with disuse, you see in the muscle reduced mitochondrial content as well as activity, so less energy production. And so all of these findings really beg the question, could there be potential alterations in metabolism in individuals with cerebral palsy? And what metabolism is it converts the food and nutrition that we take in into energy. And one sort of new emerging field that has come up as a way to assess underlying metabolism is systemic metabolomics. And essentially what that does is it enables us to assess sort of from a big picture view the overall metabolic state of an individual by looking at specific metabolites or proteins and molecules in a patient's blood that can signal potential alterations in metabolism, or the way that we use that food and convert it into energy. And no study to date has really looked at systemic metabolomics between individuals with cerebral palsy compared to typically developing individuals. And so here from this banner that has been covering my slide, essentially it was the goal of our study was to systemically assess for potential differences in the metabolic state of adolescents and young adults with cerebral palsy compared to typically developing individuals. And so overall, we obtained serum samples from 38 individuals, 19 with cerebral palsy and 19 typically developing individuals. And in order to meet inclusion criteria for the study, participants had to be between the ages of 13 to 30, able to ambulate with a gross motor function classification scale of 1 through 3, and could not have had a prior lower extremity surgery within 6 months of participating. Additionally, in the typically developing group, individuals could not endorse any history of prior or current knee or hip pain. And what we essentially did was we drew blood samples from all of our participants through an IV. We spun it down through a centrifuge and separated the red blood cells from the plasma or serum. We stored that plasma and then ran it through a mass spectrometry analysis, which basically can tell us differences in relative abundance of we looked at 261 different metabolites. And we were able to assess those metabolites for potential alterations when we compared the CP group compared to the typically developing group. And we also obtained functional measures by looking at the time that it took to walk 10 meters to rise from a seated position. And we also used a Biodex dynamometer, which assessed lower extremity knee flexion and extension strength. And we correlated those functional outcomes with metabolite levels to see if there are any meaningful trends. And so here on the left, you can see the demographic breakdown of our participants, who were quite similar, aside from the cerebral palsy group being slightly younger with an average age of 19 years compared to 20 years in the typically developing group. And in the CP group, there were about approximately 2/3 of the cohort were GMFCS 1 and 2, and predominantly spastic type CP. And on the right here, you'll see a principal component analysis. And essentially what we did was we took the 261 metabolites across 38 participants, lots of variables there. And what we did was we condensed that into fewer variables to be able to assess, are there general differences between the two cohorts of CP versus TD. And you can see here in red is the cerebral palsy group, and in green is the typically developing group. And you notice how the data points, and each data point is a participant, they tend to cluster based on those groups. So what that essentially tells us is there are sort of big picture from a 10,000-foot view, there are significant alterations or differences between the metabolic profiles in these individuals. And on the left here, you'll see a metabolite set enrichment analysis, which basically, from a big picture, looking at specific metabolic pathways, indicated to us that there were certain metabolite sets that were altered in cerebral palsy. And on the right, with our pathway analysis, we look specifically at individual metabolites. And just to orient you, on the bottom here, you'll see on the left CP1 to CP19 were our cerebral palsy participants, and then TD1 to TD19. And you can see just grossly, based on the color difference, the relative abundances were pretty stark in terms of the contrast of metabolite levels. And in particular, you can see on the right-hand side at the top, spermidine and spermine were two metabolites that were relatively increased in the TD group, whereas the vast majority of the other metabolites were relatively increased in the CP group. And we also conducted a volcano plot analysis, which essentially helps us to visualize the magnitude of difference another way, with metabolites in red representing those that were significantly increased in CP, and metabolites in blue representing those that were significantly decreased. So you can again see spermine and spermidine in blue on the top left were significantly decreased in cerebral palsy, and then all of these metabolites in red were significantly increased, with those located in the upper right-hand region representing those that were most significantly different. And so when we took a closer look at the metabolites in that upper right-hand region, you can see here in blue is cerebral palsy, and in red is typically developing individuals, and you see for each of those metabolites the significant difference in levels, but they also fell into a number of subcategories, including essential and conditionally essential amino acids, metabolism breakdown products, and TCA cycle substrates, which I'll discuss in a little bit. But essentially, this gave us some potential indications as to underlying pathways that could be altered in cerebral palsy and leading to the trends that we were seeing in these metabolite levels. And when we analyzed the metabolites in relation to functional capacity, we found that valine was moderately correlated with function, such that higher levels of valine were associated with greater functional capacity. So you can see the graphs here on the left. You see valine in that vertical axis, and then you see that higher levels of valine were associated with greater knee extensor and flexor strength, and was also associated with faster 10-meter walk times, as well as time to rise from a seated position. And similar trends were also observed for a number of other metabolites, with each metabolite being associated with greater function for a specific functional measure. And importantly, in the TD group, there were no significant metabolic correlations to functional measures. And so overall, our findings demonstrate that the metabolic profiles in individuals with cerebral palsy is significantly different than that in typically developing individuals. But it is important to note that this represents a difference in abundance, but our data cannot definitively tell us whether this is due to particular underlying underlying causes of altered metabolism. But what we can hypothesize based on our current understanding of the metabolites that were most significantly different, which I'll walk us through here, as to potential pathways that could be altered in cerebral palsy and have significant clinical implications down the road. So first, on the top left here, valine is a branched chain amino acid. It's involved in protein synthesis for muscle growth and can also be broken down from muscle cells for energy use and yield 2-ketoisovaleric in the process of breaking down muscle. And so relatively higher levels of valine and 2-ketoisovaleric acid in cerebral palsy could signify increased muscle breakdown, increased energy expenditures, or decreased branched chain amino acid breakdown. Tryptophan is another essential amino acid like valine. It is broken down via 2 different pathways, one of which is the kynurenine pathway. And the kynurenine pathway has been shown to be upregulated in skeletal muscle following endurance exercise. Thus, increased tryptophan, increased kynurenine, and increased kynurenic acid that we saw in cerebral palsy could be a sign of increased skeletal muscle activity and energy expenditure in cerebral palsy, as would be seen with endurance exercise. And histidine is an essential amino acid, and it's involved in the synthesis of carnosine. And essentially what carnosine is it works within the cell to prevent the buildup of toxic metabolites that can cause impaired skeletal muscle function and increasing skeletal muscle fatigue. And so higher histidine levels in CP could underscore lower carnosine levels and lead to increased skeletal muscle fatigue and impaired function in cerebral palsy. On the bottom left here, you'll see these metabolites, which were the TCA cycle substrates. And the TCA cycle is what occurs within the mitochondria to generate energy. And so seeing higher levels of these TCA cycle substrates could be a sign of increased energy expenditure and an increased need for energy generation in the mitochondria. And creatinine is something that you may have heard when you go for your annual visits and you get your labs drawn. It's a marker of kidney function, but it also is directly broken down from muscle. And so increased creatinine in cerebral palsy could be a sign of increased muscle breakdown. But it is important to note that individuals with cerebral palsy have also been associated with higher rates of kidney disease, and so that could be a potential confounder for these levels that we saw. And then spermine and spermidine, which is the one that I pointed out earlier, their exact role is still under investigation, but current animal models have shown anti-breakdown properties in skeletal muscle with spermidine. And spermidine is believed to be involved in the activation of autophagy. And what autophagy is the body's cell housekeeper for damaged organelles, so like the contents within the cell, that can build up and cause accelerated muscle atrophy or age-related muscle atrophy. And so autophagy helps to prevent that from happening. But spermidine and autophagy have both been shown to decrease with aging. So what we see from our study is that the decreased spermine and spermidine levels could be a sign of decreased autophagy, leading individuals to be predisposed to premature aging and muscle atrophy. And then lastly, when we correlated the metabolites to functional outcomes, we saw that higher levels of these metabolites were associated with greater functional capacity, and this could point to the possibility of the importance of these metabolites, and specifically many of these amino acids for movement capabilities in cerebral palsy. And so overall, I think our findings really have significant clinical implications down the road, because when we see sort of the general trends from our findings, we see increased skeletal muscle breakdown, increased energy expenditures, and we already know, as I had mentioned earlier, that in individuals with cerebral palsy, they tend to have inadequate nutritional intake to meet the demands that their body requires. And so our findings could have significant implications to optimize nutrition to meet the increased energy demands, as well as promote functional longevity in cerebral palsy. And certainly there were some limitations to our study. Of note, 15 of the 19 participants were higher functioning individuals with cerebral palsy, which could alter or skew the functional outcomes as well as some of the metabolic trends potentially that we saw. And as I had mentioned earlier, these were systemic metabolites, so we drew them from the blood. But again, they can only tell us differences in abundance or concentration as opposed to specifically what is going on at the tissue level with different metabolic pathways. And then lastly, these were non-fasting serum samples, which some of the metabolites, particularly the essential amino acids, are exclusively obtained in the diet. So that could potentially be a factor that could confound the findings that we saw in some of these metabolite levels. And so clearly, I think our work would encourage further investigation into sort of the underlying pathophysiologic mechanisms that could be contributing to these metabolic trends that we observed in this study. And so there are many people I'd like to thank, but I especially like to recognize the amazing participants and families who took part in our study, and as well as Dr. Jayabalan and Dr. Dayanidhi and the rest of our team for their incredible work on this project. So